urine, the basic requirement for the formation of the concentrated urine are there has to be hyperosmotic renal medulla, and the secretion of ADH plays an important role. Hyperosmotic renal medulla. Increasing osmotic gradient that is present along the medullary interstitium is produced by combined action of loop of Henle and vasa recta as a countercurrent multiplier and countercurrent exchanger respectively. The countercurrent system is defined as a system in which inflow runs parallel to and in close proximity to outflow in the opposite direction. Thus, countercurrent system has two components, that is countercurrent multiplier system and countercurrent exchanger system. Countercurrent multiplier system. The loop of Henle functions as a countercurrent multiplier and plays an important role in producing hypertonic medullary interstitium. Ascending limb and descending limb are parallel to each other and in close proximity. The descending limb is highly permeable to water, so water moves from the tubular lumen into medullary interstitium, and it is not accompanied by sodium. The medullary gradient is such that the interstitium becomes more and more hyperosmolar in the deeper parts of the medulla, which is the main reason why water flows from the lumen into interstitium and not in reverse direction. As a result, the tubular fluid becomes more and more concentrated as it descends down the thin ascending loop of Henle. The concentration will be around 1200 to 1400 milliosmoles per liter at the apex of loop of Henle. Then tubular fluid reaches the ascending limb. The thick segment of ascending limb vigorously reabsorbs sodium along with chloride and potassium. But this segment is impermeable to water. Therefore, the tubular fluid loses sodium, chloride and potassium but no water. This means the tubular fluid begins to become diluted in ascending limb of loop of Henle. Because of the movement of solids from ascending limb of loop of Henle to interstitium, the osmolarity of medullary interstitium increases and it becomes hypertonic. This situation is maximum in GJ nephrons because of long loop of Henle. The hypertonic tubular fluid is then delivered to the DCT. In the early part of the distal convertible, reabsorption of the sodium continues, which causes further dilution of the luminal fluid. Finally, the fluid enters the cortical collecting tubule. When ADH starts in cortical collecting tubules, water flows massively from CD to the interstitium through aquaporins. This flow of fluid continues until the osmolarity of the collecting fluid attains that of interstitium. Thus, the urine formed is very concentrated. The medullary interstitial hypertonicity is also contributed by urea, which moves out of the inner medullary portion of the collecting tubule. This urea also plays an important role in the concentration of urine. This in turn is influenced by a protein diet. So, high protein diet produces high concentration capacity of kidney. So, this is the diagram showing the nephron where it is showing concentration of urine in different parts. So, when the fluid enters, filtered tube enters into the descending loop of Henle, water is highly permeable, and because of the movement of the water, the tubular fluid becomes concentrated and it reaches almost 1200 to 1400 milliosmoles per liter at the tip of the loop of Henle. And when the fluid comes and reaches to the ascending thick loop of Henle, where the sodium, chloride and potassium are reabsorbed and water is not reabsorbed, the solution becomes hypotonic, and by the time it reaches to the distal converted tubule, so it is hypotonic. And when the fluid reaches the collecting duct, because of the concentration gradient at the interstitial medullary, the water is going to be reabsorbed, and again the urine gets concentrated, and concentrated urine get excreted in the concentrated urine get excreted. See, here you can see the isotonic plasma enters, as and when it reaches to the tip of a loop of Henle, it reaches the concentration of 1200 to 1400, and because of the reabsorption of the solutes like sodium, chloride and potassium, it again becomes hypotonic. But when it reaches to the collecting duct, again lot of the water is reabsorbed, and this hypotonic fluid becomes hypertonic, and it is excreted by the kidney. The next figure shows recirculation of urea absorbed from the medullary collecting duct into the interstitial fluid. This urea diffuses into the thin loop of Henle and then passes through the distal conduit tube and finally passes back into the collecting duct. The recirculation of urea helps in trapping of the urea in renal medulla and contributes to the hyperosmolarity of the renal medulla. So thus the reabsorbed urea from the collecting duct, it is going to enter into the loop of Henle. It gets absorbed, so again it is secreted or reabsorbed back only in the collecting duct, and thus the recirculation of the urea is going to help in the development of the inner medullary concentration gradient, and thus it is going to help in the concentration of the urine. The heavy dark lines from the thick ascending loop of Henle to the medullary collecting ducts indicate that these segments are not permeable to urea. That is, the numerical values have been given in moles per liter of urea during antidiuresis when large amount of antidiuretic hormone are present. The percentage of filtered load of urea that remain in the tubules are indicated in the boxes. Okay, so you can see here. So this is how the urine medullary concentration gradient is maintained at the outer medulla. I mean the interstitial, the tonicity is around 300 milliosmoles per liter, and at the depth of the loop of Henle, it reaches almost 1200 to 1400 milliosmoles per liter. Countercurrent exchanger system. Vasa recta functions as a countercurrent exchanger. It is responsible for maintenance of hyperosmolarity of medullary gradient developed by countercurrent multiplier system. If blood vessels were straight and the blood flow were high, the solute pumped into the renal medulla by countercurrent multiplier system would rapidly dissipate. The vasa recta also has a descending and ascending limb. It runs parallel to loop of Henle. Both limbs are permeable to water and solute except for the plasma proteins. Blood entering the distal loop has an osmolarity of 300 milliosmoles per liter, and the osmolarity of the blood leaving the loop of Henle is or the ascending loop is around 325 milliosmoles per liter. As the blood descends into the medulla, it becomes progressively more concentrated and partially by solute, and entry from interstitium and partially by loss of water into the interstitium. By the time blood reaches the tip of the vasa recta, it has concentration about 1200 milliosmoles per liter. As the blood ascends back towards the cortex, it becomes progressively less concentrated as solutes diffuses back into the medullary interstitium, and as the water moves into the vasa recta. The blood passing through the ascending loop of vasa recta carry very little amount of sodium chloride from the medulla. Thus, sodium chloride and urea are exchanged for water between the ascending loop and descending loop of vasa recta. Hence, this system is called as countercurrent exchanger. If vasa recta was straight, sodium and urea in the interstitium would have been removed by circulation. The sluggishness of blood flow ensures the retention of sodium in the medullary interstitium. Okay, so this is the descending loop of the vasa recta, where progressively as the fluid reaches, the sodium is going to enter from the medulla into the loop, and the fluid gets the blood gets concentrated. By the time it reaches the tip of the loop, the concentration is around 1200, and while the blood when it is ascending in the loop of recta, the again the sodium chloride and urea, it gets reabsorbed and water is going to enter inside because of the concentration gradient. Okay, and again the fluid becomes hypotonic. Secretion of ADH. In the late distal tubule and cortical collecting tubules, the osmolality of the fluid depends on the level of ADH. With high levels of ADH, these tubules are highly permeable to water. Formation of dilute urine. When water content in the body increases, the kidney excretes dilute urine, and it is achieved by inhibition of ADH secretion. And this diagram shows the formation of the dilute urine when ADH levels are very low. So ADH is mainly going to act on the collecting duct, and when in absence of ADH, the water is not going to be reabsorbed back. So the excess amount of water is going to excreted, and it is going to form the large amount of dilute urine, and that is how ADH controls the amount of urine or osmolality of the urine.